risk of having embolization, I think.
Exactly.
Thank you both. All right. I think our next speaker kind of pulls it all together there with what is happening at the endothelium. So talking about endothelial dysfunction, Dr. Walid Kadro, Director of the Golden Center for Advanced Cardiovascular currently, and going to talk about endothelial dysfunction in COVID. Dr. Kadro, take it away. And you need to unmute yourself, sir.
Can you hear me now?
All right, now. We cannot hear you very well.
Can you hear me now?
That's better. Can you increase that volume somewhat?
Okay.
We're all the way in Texas, so we need a little volume.
Yeah. Can you hear me now?
Yes.
Yeah, okay.
Yes.
Let me share my screen now. Okay, good evening, everybody. Let me thank all the organizers, especially Dr. Halak and Dr. Krager for the nice invitation for this nice symposium to cover about COVID-19. I'm happy that I'm taking off my hat as an interventional cardiologist and putting a new hat as a vascular biologist, and we'll try to cover the topic of endothelial dysfunction. Just let me put on the slideshow. Okay. So everybody knows about the endothelium, about the vessels, and every vessel has adventitia and media and has the endothelium. And you know the endothelium is only one layer, but has a lot of function. So the main function of that, apart from protection and maintaining the good flow, it works as an anticoagulant and prevent the formation of in situ clots, and also work as a vasodilator through the production of an important substance called nitric oxide. And the nitric oxide is really synthesized in the endothelium by converting the arginine to citrulline and adding oxygen, and thus create the nitric oxide. And the nitric oxide has a lot of important function. It promotes vasodilation, it increases arterial compliance, and cause positive remodeling and dilation of the vessel, and also inhibit a lot of bad things like smooth muscle proliferation, and prevent apoptosis of the endothelial cell, prevent thrombosis, and prevent platelet activation. It also prevent the secretion of the tissue factor when it's intact. But however, when it's the endothelium is really damaged, it releases tissue factor. Also, the nitric oxide prevents inflammation and production of cytokines. So this is the effect of the nitric oxide production through the enzyme called endothelial nitric oxide synthase. And after the production of nitric oxide, it has a lot of good things that promote laminar flow, prevent adhesion of all leukocytes, and prevent activation of platelets, and also work through cyclic GMP for production of the substance that cause smooth muscle relaxation. And with that, this is the one of the tests that I used to do in the CAS lab when I was a fellow, and that's this is the baseline, and you see that the vessel is not dilated by really giving acetylcholine infusion, which work through production of nitric oxide, cause significant dilation of the circumflex artery. However, if the nitric oxide is not really bioactive due to damage of the endothelium, we'll get all of those things inactivated. So we are not preventing adhesion. So there is more leukocyte adhesion. There is less oxygen delivery. There is increased platelet activation, and there is no vascular muscle effect. And with that cause vascular muscle contraction and spasm of the vessel. And really, the endothelial dysfunction predict cardiovascular mortality. This is a small study that evaluated the patients who were going for vascular surgery, and they did endothelial function through the brachial artery ultrasound, and found that the people who has normal vasodilation in response to acetylcholine has almost fourfold protection from cardiovascular event compared to the people who has really a bad response to dilation. And there is too many studies, most of them are really positive that endothelial dysfunction predict cardiovascular event. So how do we get endothelial dysfunction? And the most important thing is the oxidative stress, because if we have really a superoxide and active oxygen species, reactive oxygen species, when it really work and interact with nitric oxide, it produces this substance which is nitric superoxide, and this chemical activation is really quite fast, happen like a diffusion, and the rate constant is really high. And a lot of things cause this endothelial dysfunction and impair nitric oxide by activity. Everybody knows about hypercholesteremia, smoking, diabetes, hypertension, and coronary artery disease. Now, how this can we interact between the reactive oxygen species and nitric oxide? We know nitric oxide causes vasodilation, platelet inhibition, smooth muscle quiescence, and vasodilation, normotension, and prevent proliferation. However, if we get reactive oxygen species, we get vasoconstriction, thrombosis, smooth muscle hypertrophy, and hypertension, and endothelial cell damage or death. And we know they work against each other. Now let's talk about reactive oxygen species. And we know that this oxygen, regular oxygen, if we add an electron to it through an enzyme, which is quite common in the white blood cells, the enzyme called NADPH oxidase, and that's produced really superoxide. And let me put this red dot to indicate that's really a radical and a harmful substance. And if we add another electron, we'll get the hydrogen peroxide. And if we add another electron, now we'll get reduction and we'll get the another really harmful called hydroxy radical. And finally, if we add another electron, we'll end up with the harmless substance of water. Now, how our body protect us from that? There is a lot of things. Now we have an enzyme called superoxide dismutase, which has 3 subunits, one in the mitochondria, one in the extracellular matrix, and one inside the cytoplasm. And the cofactors for one is zinc, the other cofactor is copper, and the third cofactor is manganese. And this enzyme take this superoxide and bring it back to oxygen and hydroxyperoxide, which is less toxic substance, but still it works as a killer in the white blood cells. Now, the other mechanism...
Dr. Carter, 2 minutes, please.
Yeah,
2 minutes.
The other mechanism are really the substance of glutathione, and if reduced form, it's really converted to an oxidized form, and that causes this hydrogen peroxide to be converted to water, and through an enzyme called glutathione peroxidase. Now, this can recharge again by another reducing enzyme called glutathione reductase. Now, with that, also there is another prevention mechanism through an enzyme called catalase, which really take this hydrogen peroxide and convert it back to normal oxygen and back to water. This is the normal things. Now, what's happen if we get increased oxidative activity in obesity, hypertension, in type 2 diabetes? Now we'll go back to what happened here in our COVID patient. We have oxidative stress, and we reviewed that, and let's concentrate on this. And we'll find that now there is no protection because of the exhausted effect of the superoxidase dismutase and this glutathione peroxidase. And what will happen, we know that there is angiotensin 2, and angiotensin 2 is really converted to good substance through an enzyme ACE2. But however, this angiotensin 1,7 also work to suppress the nitric NADPH oxidase and prevent the production of this bad substance, superoxide radical. However, if we get the SARS-CoV, yeah, this is the last slide. If we get the SARS-CoV-2, we'll get more inhibition of this ACE2, so there we have now double inhibition. That's mean increased activity of NADPH oxidase and increased production of superoxide radical and increased oxidative stress. So this is the summary slide of hypothesis, how we get this bad coagulation. We get SARS-CoV-2, and that will get decrease in ACE2 activity after the connection, virus interact with this receptor, and this will get increase of angiotensin 2 and decrease of angiotensin 1,7, and that's lead to increased oxygen, reactive oxygen species, and this radical substance, and that's cause endothelial cell dysfunction, and with that we'll get increased production of bone volume factor from this damaged endothelial, and that will end up with this thrombosis. And that's the mechanism of endothelial dysfunction and why we get increased thrombosis with COVID-19. I have a lot of studies, but I think I did reach you the message, and thank you very much. I'd be happy to get any question.
Thank you, Dr. Khadro. That was amazing. We all needed to remember about all these pathways and endothelial dysfunction. Unfortunately, we have no time for questions right now, but later, hopefully after we finish all of this. Thank you so much, sir. Our next speaker is Dr.